of them. So, our first presenter is going to be Dr. Marilor Yasbo, and she is the founder and chief scientific officer of Alacris Theranostics. Marilor is joining us from Germany this evening, and I know I've seen in the chat that we have several folks that are outside of the US, which we do every time. But for our presenters, when they end up having to give a pitch in the middle of the night, I believe it's 2 a.m. there, we appreciate you being here even more.
So good afternoon. My name is Marilor Yasbo, CS1 and founder of Alacris Theranostics, an innovation leader in personalized oncology. And thank you for giving us the opportunity to present our flagship product, the CMTA, which stands for Comprehensive Molecular Tumor Analysis, which is a high-precision diagnostics for cancer patients, filling a crucial need in oncology. So the problem with current oncodiagnostics is that every tumor is unique, but it's unique, but it's also a complex world of phenotype, which is the result of a mesh of molecular alterations. And the problem really with the current test is that they are unable to capture this, the tumor landscape. And why is that? It's because those tests are DNA based and can identify mutations, but miss a number of events which happen in the tumor, including the expression status of DNA mutations, the gene fusion, RNA splice variants, dysregulated oncogene, prognostic gene signature, and tumor microenvironment with the immune infiltration, which all together, collectively or individually, represent targets of potential treatments. And so the only way actually to access those events is to read out the RNA molecule, which is a functional readout of the DNA. So the issue is that by the current test, the oncologists are missing true option for personalized therapy, which might lead to ineffective treatment, drug resistance, side effect, wasted money also. So there is a gap in basically reading out what the tumor is about. So the CMTA solution that we propose is a comprehensive sequencing-based diagnostics which uncovers the tumor behavior context. And how is that? So we integrate to the DNAsec, to the DNA information, the RNA-seq, which is a messenger information, the functional message, which give access to altered mRNA abundance, which picks up overexpressed oncogenes, splice variant, gene fusion, immune infiltration. And with this data, with this information, we use our computer programs to chart the nodes of the cancer pathways to be able to match the therapies, potential therapies, drugs to those nodes and deliver to the oncologist the CMTA report. So the benefits really for the oncologist and for the patients is to have a personalized tumor data report which provides more actionable targets, allowing to have treatment recommendation which guide clinician choice based on the tumor of the patient's data and not on guesses. So that offers new possibilities, extended possibilities for the treatments. So how that works? Basically, we extract the signal from the noise to tease out the key tumor information to get there to those to the drug targets. And so how it is that we achieve this and ahead of most companies, I shall say, it's simply because we have been working years on the issue of RNA-seq and analyzing RNA-seq. So there are 4 crucial steps. So first, we have been pioneers in protocols of single-strand RNA-seq, which provides a level of precision that we need for these diagnostics. The second step is that we have built integrative analysis, which allow to read out measures, quantities of RNA-seq and integrate with the DNA information. And with that big data at the step 3, we are able to see really the important events for each individual tumor. So to have a personalized landscaping of the tumor, which is used as a basis for the drug recommendation. So that involved a number of events, as you see, which can happen and happen in general in the tumor. Some of the important ones are really the expression level of all genes. We can measure this here. You can see that, for instance, in that particular tumor, VEGF-A, which is a angiogenic factor, which is targetable, is very high compared to the mean. We can classify the tumor and read out the composition of the immune infiltration. And the step number 4 is to be able to automatically control this together in a personalized tumor report system, which will support also combination therapies, listing a number of those alterations and the matched drugs. So that is really the breakthrough here. So how does it validate? Does it really help to get more alteration and the drugs that match to it? Yes, it does. There have been several trials and translational studies which have been evaluating how this approach really boosts the possibilities to choose targeted therapy, and they all agree, including our study, to that is more than 30% increase in the number of cancer patients receiving a targeted therapy guided by those type of integrating approach. Sorry. So I showed an example, a case example, here on the right-hand side from our study. It is a skin melanoma which didn't have a BRAF mutation. We detected 150 mutations in it, but none of those mutations were really relevant. What was relevant is this NTRK3 gene fusion here, which is targetable. We identified this, and the clinician went ahead with the recommendation to treat with entrectinib. And as you can see here, that was a metastatic case. So we see 3 metastases, which really reduced after a few months of treatment. So that is really a test. The CMTA product is, I could say, universal, because we can use it for any type of tumor. And the timeline of our product, our company, I could give a short glimpse very shortly. We started the company with a strategic investment of 2.5 million from Kiagen, and started to develop the CMTA product. We received the German Innovation Prize, and kept going with for quite a number of years with R&D grants from European Community and the German government for 15 millions. And during that time, we performed, the product was further developed, optimized. We introduced the CMT in the market, which partial insurance reimbursement for some entities, and introducing in other countries now. One very important point, which is one milestone, is ISO 15189 medical lab accreditation for the CMTA. And we are now at a stage where we we are seeking for serious funding to ramp up our capacities and develop further in the marketing of this product. So who is Alacris? Alacris team leadership. We are basically 4 main people, Hans Lehrer, George Church, myself, and Bodo Lange, who have long expertise in pioneering genomic technologies, which allowed us to develop the CMTA and the predictive modeling of drug response. And on this, I think that I can stop here and take questions. Thank you very much for listening.
Wonderful. Thank you so much for being here tonight. We're excited to have you for sure. And I know um, this technology, so from what I understand of your company, this technology is not new technology. It sounds like these technologies are all available. So, one question I would have is what's to prevent someone else from doing the same thing? And tell us a little bit more again about what really differentiates you from your competitors.
Yes, that's true. The technology is out there. We published those protocols and patents them long time ago. What prevents others to use it is the experience. We have been pioneered in these RNA-seq technologies, so we started ahead. We had a head start and spent a lot of time in this analysis. The reason is that other people don't use it is that the world has been and is still dominated by the DNA test because it's easier. It sells well as well. It still sells well, and a number of people were not companies were not really in the swing of things to get going with the analysis of RNA-seq. So it's a lot of experience, of know-how. It's there. I totally agree with you, but it takes a long time experience to know how to deal with it, to be able to have a quantitative measure and to be able to turn it into a diagnostics, because you have to be sure of what you are doing there. So that's why we are very happy that we got the medical accreditation for this combined test using the RNA. So we are ahead of our competitors, even the large companies, because of this know-how, because we have learned how to interpret the data and to analyze it, yes, with the right tool. There's the analysis and the filtering of the events. So this is simply know-how.
That's fantastic. And before moving on to our questions from the audience, I noticed on your timeline you had something that was called the cup test. Um, I'm assuming that's maybe a future product. Can you tell us a little bit more about that?
Yes, the CUP test is also something that has been around. So CUP stands for cancer of unknown primaries. They are up to 5%. In Europe and the US. In other countries like India, they're up to 25% of cups to be analyzed, and we have developed this cup test to be able to give a name to the tumor. So as the cups have unknown primaries, so we don't know where they come from initially, from the pathology, from simply looking at the tissues, sorry, as a tissue slide, but with the molecular analysis, we can place the tumor and determine where it comes from, which is closer to a, let's say, colon cancer or breast cancer, and we can also read the mutations from the RNA-seq read. So this is a product that we are launching now. We start with India, and we will distribute it as well in other countries.
So again, probably the ability to treat these cancers more effectively, I'm guessing, even though you may not know where the primary cancer was.
Yes, but with the analysis, you know, because with a gene expression signature, we are placing the cup, when we get a cup, to the closest known cancer, because we have a number of signature for known cancer entities, and then we match the cup to its closest neighbor, so that we can then crack the actual origin of this cup, and that changes the treatment also, because we can also read mutation and fusions and so on. So otherwise the cups are treated almost exclusively with chemotherapy, and that gives a chance to the cup to have targeted therapy when it is possible.
Sure. Dr. Macaluso is in our audience, and he's curious to know, can this test be used on all types of cancer or only certain types of cancer?
all types of cancer, because the CMTA is based on sequencing the tumor, getting the RNA and DNA informations, and listing the alteration we found and matching the drug. So any type of cancer can enter into the analysis.
Wonderful. And Dr. Desai, who is an oncology fellow at Mayo Clinic, has a question for us. How is your technology different from Tempus/Foundation, who also report RNA data, although for research purposes only, it sounds like? What is the depth of sequencing in terms of number of genes, etc.?
Yes. Yes, it is true that Tempus and others also propose partial analysis of RNA-seq. We analyze data from Tempus from some of our clients, so we know how the data are. They also have different protocols. So we analyze with our technology, we analyze all the genes. So we have the 20,000 genes, not all, or 25,000 human genes. Not all are cancer genes, but we have them all in the analysis. This is how we can read the microenvironment as well, which are the immune cells in and around the tumor. So what differentiates us from Tempus is sometimes Tempus and other companies use a technology for the RNA-seq, which is they use capture, and that basically kills the dynamic range. So it's much more difficult to measure the quantities of the molecule. So if you are not quantitative anymore, it's very difficult to say if a gene goes up and down. But the main difference of with all of our competitors is the analysis and the reporting. They do not report the tumor microenvironment in the details that we are reporting, in neither the change of gene expression. We also report splice variants and so on. They are not getting into that.
So it sounds like your analysis and your report is much more comprehensive than what your competitors are doing.
Yes, that's it, because they had a different strategy, right? We have developed that over years. You saw on the timeline, it's a 10-year story. We were also part of the TCGA, ICGC for Europe enterprise. So we had a long learning curve as well, how to tweak, not to tweak, but to get the information out of this data. So that's really the main difference. It's much more comprehensive, it's more quantitative, and the report is more informative.
Wonderful. And you're in market like some of these others, which is fantastic. Well, Marilor, thank you so much for being with us tonight. We appreciate you sharing your company with us. I think it's very exciting in the world of oncology treatment. A lot more cancer patients probably, again, are going to have successful treatments just by identifying those additional targets that you're able to identify. So thank you for all the work that you do.
Thank you.